So welcome back to the second part of our lecture series on the functional anatomy of neurons. And in this lecture series, we will talk a little bit more about the organelles within neurons. Like any other part of the body, neurons, the nervous system is made out of cells. And neurons contain, like all the other cells in the body, organelles within the body. That includes the nucleus, the rough endoplasmic reticulum, the Golgi apparatus, mitochondria, lysosomes, and the cytoskeleton of the cell. In neurons, because they are such specialized cells, some of those cell components play a particularly important role. And for that reason, I would like to introduce you a little bit to those organelles and the particular importance of some of those organelles within neurons. So first of all, neurons, because of their long extensions, because of their long neurites, have a very elaborate cytoskeleton. And the cytoskeleton in neurons is particularly important for stabilizing those long neurites and also for transport of proteins and other molecules inside of those neurons. So the cytoskeleton is an intracellular network of proteins, and these are protein fibers, and they consist out of the biggest ones that are the microtubules. They are shown here in green. Then the medium-sized ones, and these are intermediate filaments. In neurons, they are called also neurofilaments, and these are indicated here in orange. And those intermediate filaments are very important for the diameter of the neuron, because those side arms of those intermediate filaments, they repel each other. And for that reason, if you have more of those intermediate filaments that repel each other, the diameter of the neuron gets bigger. And then we have the actin filaments, and the actin filaments play a very important role in the stabilization of the neurons, in cell adhesion of the neurons, and in development of neurons. Here you can see a higher magnification of those three filaments that make up the cytoskeleton. And what you can see here, the actin filaments, consist out of globular subunits, and they're indicated here as these little red balls. These are globular subunits, and they are put together in a way that they form two protofilaments, two strands that make up one actin filament. And important to understand here is that these actin filaments are very dynamic structures. They assemble and disassemble on both ends, and for that reason can grow into one direction and can help the neurites, the dendrites and the axons of the neurons to grow out in one direction. And as you can see here as well, if you think about, you have a number of those fibers, they also stabilize the neurons in the cytoskeleton of the neurons. Then the intermediate filaments, they are about 8 to 10 nanometers in diameter, whereas the actin filaments are only about 6 to 8 nanometers. This goes down to electron microscopic sizes. These intermediate filaments, again, are very important for the diameter of a neuron. They're not made up out of globular subunits, but out of a mixture of different proteins, and they're very stable, and they stabilize the neurons within those neurons. And then the largest fiber in the cytoskeleton are the microtubules, and the microtubules are about 25 nanometers in diameter, and they consist out of, again, globular subunits, very similar to the actin filaments, that can assemble and disassemble very easily. And for that reason, they are highly dynamic structures. However, this assembly and disassembly of the microtubules is at a cost. So it costs energy for the cell. So if the microtubules are assembled and disassembled, the cell needs to put energy into this. And for that reason, in mature neurons, when the long axons are formed and the dendrites, and I have an example here of bipolar neurons. That is an example of an invertebrate system. And these are sensory neurons that perceive touch in invertebrates. You can see see those very long dendrites and those axons that come out of here. And you can see this here. So these are these very long dendrites, and the axons are down here. So if they need to be stabilized in mature neurons in order to downregulate the energy demand that the neurons have, there are so-called capping proteins that attach to those microtubules, also to actin filaments. And those capping proteins stabilize the microtubules and prevent them from disassociating and associating all the time. So they take a little bit of this dynamic out of the microtubules. And for that reason, in mature neurons, stabilize those dendrites, which is critically important. Microtubules are also very important with regards to transport of proteins and molecules along those long neurites that the axons have. We've talked a little bit about the synaptic junctions at neurons that they form. And as you can see here, there are a lot of vesicles and there are a lot of receptors and proteins that are required for those synapses. All those proteins and molecules that are present at those synapses and also in the membrane of neurons need to be transported to these areas. And this transport takes place along the microtubules. So the microtubules are basically highways within the neurites, along which those proteins that are packaged in vesicles in the Golgi apparatus, and we'll come back to this in a few slides. And then those packages are, with the help of particular proteins that attach to those microtubules, transported either in one direction or in the other direction. So if proteins are transported from the soma to the axon terminal, we call this anterograde transport. If proteins are transported from the axon terminal back to the soma, we call this the so-called retrograde transport. So anterograde transport is from the soma to the axon terminal, from the soma to the axon terminal. Retrograde transport is from the axon terminal back to the soma, where the metabolic end products that are being transported back to the soma can be processed and either released by the neuron or metabolized and recycled within the neuron. So another organelle within neurons, and that most cells have this, is the nucleus. The nucleus contains the genetic material in form of DNA, deoxyribonucleic acid. And it consists out of a number, so out of chromosomes, and those chromosomes contain genes. And each gene can synthesize certain proteins. Humans have 46 chromosomes, and when a certain protein is made or transcribed from the chromosomes, we call this gene expression. So this takes place within the nucleus. And this gene expression takes place with the help of an enzyme that is, or a certain protein that is called RNA polymerase. And this RNA polymerase creates messenger RNA, and you've heard probably about this, or mRNA. And this mRNA is then processed and transported out of the pores that are in this nucleus in the nuclear envelope in these that surround the nucleus. And this messenger RNA is then transported to the rough endoplasmic reticulum, where it then is recognized by ribosomes, and those ribosomes are now transcribing this messenger RNA and make the proteins out of this messenger RNA. So transcription takes place within the nucleus, is when gene expression takes place and messenger RNA is produced. Translation takes place outside of the nucleus at the rough endoplasmic reticulum or at free ribosomes that are in the cytoplasm of the cell. And you can see an example of this in this image here. So as the messenger RNA is transported out of the nucleus, it usually is recognized by ribosomes here at the rough endoplasmic reticulum, and the translation of this messenger RNA into a certain protein takes place. And this is a particularly abundant organelle within neurons, because neurons have this very high demand for proteins in form of ion channels, in form of transmitters, in form of metabolic enzymes that are very important for processes within the neurons. So neurons have a lot of this rough endoplasmic reticulum. And you can see here, this is an electron micrograph of a rough endoplasmic reticulum. You can see here membrane stacks, and those membrane stacks contain these little black dots, which are the ribosomes. And you can see in this case here, the ribosomes are on these membrane stacks, and they make proteins that are incorporated into the membranes, such as ion channels or receptor channels. Or we have free ribosomes that are free floating in the cytoplasm, and those ribosomes synthesize proteins that are not incorporated into membranes. You can see this in this image here as well. Here are free floating ribosomes that get the messenger RNA and then synthesize proteins and translate this messenger RNA into a protein. And here we have a ribosome that is associated with the rough endoplasmic reticulum that incorporates the protein into the membrane of the cytoplasmic reticulum. And this would be an example of such a protein that is incorporated into the membrane, and this would be an ion channel within a neuronal membrane. So these ion channels are very specialized, and they allow certain ions to flow through. Another organelle that is particularly important in all cells, but particularly abundant in neurons, are the mitochondria. And you might know that mitochondria are the power plants of the cell, that they produce ATP, and ATP is so crucially important for all those metabolic processes, such as assembling microtubules or actin filaments, or activating enzymes, or pumping ions within the neuron in certain directions. And we will come back to this throughout this lecture series, how important ATP really is for the function of a neuron. And if a neuron does not have energy, neurons die fairly rapidly because they don't have any energy resources. So for that reason, areas in which neurons require a lot of energy are usually laden with mitochondria that deliver the energy for all of those processes. So very prominent organelles within neurons. The Golgi apparatus is in close proximity to the rough endoplasmic reticulum, and is important for proper packaging and tagging of those proteins in order to tag them to get to their target area. And you can very much compare this with a label that you put on a package that you want to send to your grandmother. So this package has to have the name of the person whom you want to send this to on it in order for the postman to deliver this to the right direction. And this is what the Golgi apparatus does. So those proteins that are now incorporated in the rough endoplasmic reticulum are incorporated now into the Golgi apparatus, and the Golgi apparatus has two sides. It has a forming side, and it has a maturing side here. In the forming side, it's also called the cis side, or the maturing side is also called the trans side. In the cis side, the vesicles are forming, and the proteins are incorporated into the Golgi apparatus. They are processed throughout the Golgi apparatus. They are tagged the right way. And then here in the trans side of the Golgi apparatus, where the vesicles are then released, those vesicles are now tagged and transported through along the microtubules to their target organs, where they are then incorporated into the cell membrane, or where they are then in the right area where they are required, if they are not membrane bound. So to summarize this, we have our nucleus the messenger RNA is produced in the nucleus, or transcribed in the nucleus, then transported out of the nuclear envelope that surrounds the nucleus through those pores. Then the messenger RNA is translated at the rough endoplasmic reticulum at ribosomes or at free-floating ribosomes. These proteins that are incorporated in this case here in the rough endoplasmic reticulum are then transported to the Golgi apparatus, where they are then processed and tagged and now transported along the cytoskeleton, along the microtubules to their target areas. Another very important component or organelle within neurons are the lysosomes. And lysosomes contain proteolytic enzymes. And those proteolytic enzymes are able to digest proteins and turn them back into their individual building blocks. So, and those building blocks can either be reused within the neuron, or they can be exocytosed or released from the neuron into the environment as a metabolic end product. And this is then usually cleaned up by glial cells or by the cerebrospinal fluid, flushed out by the cerebrospinal fluid that we have in our central nervous system. So lysosomes have to be surrounded by a membrane because those proteolytic enzymes otherwise would digest the neuron. So in this case here, they take up the metabolic end products. In this case here, it's an old or damaged organelle, and they metabolize it and basically clean the neurons up and make sure that the neurons are maintained in proper functional condition. Microglial cells also have a process that is called phagocytosis. And phagocytosis is very much that microglia can take up waste products into the interior and then metabolize those waste products or transport those waste products away. And for that reason, the microglia are very important, basically garbage carts within the brain. They make sure that there are no debris, that there's no debris in the central nervous system. If there's some brain damage where some neurons degenerate, this is then phagocytosed by microglia and cleaned up. So this is our little tour through the organelles, and we will, in our next lecture series, talk about the action potential.